Hello, PsychU community. This is Dr. Carolyn Tyler from Otsuka Neuroscience Field Medical Affairs. I am joined today by my colleagues, Dr. Rachel Self and Dr. Heather Davidson. Welcome to an overview of the psychoneuroimmunology of mood disorders. This is an exciting emerging area of research within psychiatry, and this program seeks to provide you with an overview of the science and potential therapies in this field. This is part 2 in the psychoneuroimmunology series. If you would like a general overview of psychoneuroimmunology basics, please review part 1. Now let's review the objectives for this program. So we'd like to provide a rationale for the focus on inflammation as a contributory component in the underlying pathophysiology of mood disorders. Also, elucidate the key mediators and processes underlying neuroinflammation in mood disorders. Outline the potential causes and consequences of neuroinflammation in mood disorders, and finally discuss potential treatment modalities targeting neuroinflammation specific to mood disorders. To begin, let's review the inflammatory response in the brain. As shown here, there are several proposed mechanisms by which peripheral inflammatory cytokines may access the central nervous system and initiate local immune activation. These include passage through leaky areas of the blood-brain barrier, active uptake mechanisms of cytokines across the blood-brain barrier, direct transmission of cytokine signals through the vagal nerve, and regulation of the hypothalamus pituitary adrenal or HPA axis by glucocorticoids. For a more in-depth review of the basics of psychoneuroimmunology and the inflammatory response of the brain, again, please review part 1 in this series. There are several key players involved in neuroinflammation within the central nervous system. As shown here, these include glial cells, including microglia, astrocytes, and oligodendrocytes, cytokines, glutamate, mitochondrial and oxidative stress, the HPA axis, S100B, and arachidonic acid. Now I'm going to turn it over to my colleague Rachel Self for a review of the basics of mood disorders and inflammation.
Thank you so much, Carolyn. So as we can see with the slide shown here, and we have a fun little graphic that we've added for you as well, this relationship between mental health and inflammation is really not a new concept. It was actually first noted way back in 1887 by Dr. Julius Wagner-Jauregg out of the University of Vienna, which actually afforded him later in 1927 to have the opportunity to be the only psychiatrist to ever win the Nobel Prize in physiology. Inflammation, mood disorders, and sickness behavior, as we discussed in the previous PNI basics deck, is really an important concept. So we know that in non-depressed individuals, inflammatory stimuli can provoke this sickness behavior, which is that feeling of overall malaise, low energy you experience when you have a cold or the flu. Cytokine activity is increased. It can lead to behavioral changes that are consistent with both sickness as well as depression, such as more sleep, appetite disturbances, and fatigue. There have been studies that show inflammatory stimuli are actually present in about 45% of individuals who have depression. And if you introduce an inflammatory stimuli, you can actually create depressed symptoms in otherwise healthy individuals. Animals and humans alike can benefit from neurovegetative symptoms such as lethargy, decreased appetite, decreased mood, increased sleep, decreased interest in activities and exploration, as well as decreased sexual activity, just to allow the organism to devote time and energy to healing and protection from future attacks. And this is how this phenomenon has become referred to as sickness behavior. On a population level and from an evolutionary standpoint, it makes sense because it is beneficial to prevent the spread of infections. Although not every patient with major depressive disorder exhibits high inflammatory markers, evidence does suggest that high levels of C-reactive protein, which is one of our pro-inflammatory markers, is detected in a substantial portion of individuals with depression, with results indicating anywhere from a quarter to nearly half of patients with depression showing this high inflammatory tone. And recent data indicate that increasing plasma CRP concentrations in patients with depression is also associated with decreased functional connectivity within reward circuitry within the brain, as well as increased glutamatergic levels, which we heard about in the basics deck from Heather. And all of these can contribute and be correlated to the symptoms of depression, including anhedonia. There are several different hypotheses that have been proposed to try to explain these different interactions. And we know that the interaction between inflammation and mood disorders, including depression and bipolar disorder, are bidirectional. There's definitely a bidirectional link and a self-perpetuating component to these two. We also know that treatment success is impacted by changes in the inflammatory tone. So if inflammation can in fact induce and exacerbate sickness behavior, inflammatory comorbidities would be expected and associated with mood disorders, which is what we indeed see. And you'd see increased prevalence of mood symptoms found in a variety of inflammatory conditions, including autoimmune diseases, cardiovascular disease, diabetes, obesity, metabolic syndrome, etc. Increased inflammatory biomarkers actually predict antidepressant non-response, and inflammatory cytokines can sabotage and circumvent mechanisms of action of conventional antidepressant therapy, as well as disrupt the effectiveness of psychosocial interventions. So as we transition to the next slide, when we're talking a little bit more about the inflammatory cytokines, we see that they can also influence neurotransmitter metabolism and bioavailability. And this goes across the board for not only our monoamines, but also, as we heard in the PNI basics deck, more importantly, our main excitatory neurotransmitter glutamate, as well as our main inhibitory neurotransmitter GABA, really impacting the overall functioning of the brain in general. I'm going to now hand it over to Heather to talk to us a little bit more about the specifics in inflammation as it relates to patients with major depressive disorder.
Thank you, Rachel. So what I'm going to do now is walk you through some of the studies that Rachel has kind of alluded to in her overview, specifically in major depressive disorder. So I'll touch on how inflammation affects key neurotransmitters and show you some data for that and how it might be useful in predicting the treatment response, although we're not quite there yet, but I'll kind of describe that in more detail. So what you're seeing on the screen now is on the left-hand side, C-reactive protein, as Rachel mentioned, is produced by the liver in response to inflammatory cytokines, and most notably in response to interleukin-6 and tumor necrosis factor, which are two cytokines involved in the inflammatory response. It can be measured by taking a blood sample to determine if peripheral inflammation is present, but it doesn't necessarily tell you which organ system is affected. So in the study that's shown on the left, you see the results of a principal component analysis from patients with major depressive disorder, showing that high C-reactive protein levels in the plasma correlated with high levels of inflammatory cytokines in the cerebrospinal fluid, which tells us that the inflammatory signal is crossing the blood-brain barrier. In addition, there was also a correlation for inflammatory cytokines and C-reactive protein with depressive symptoms, and the correlation was primarily for reduced motivation and anhedonia. As Rachel had mentioned, these are similar to what we are thinking of when we talk about sickness behavior. On the right-hand side of your screen, you see the results of another study, and it really makes an important point that Rachel has already touched on. Not all patients with major depressive disorder will have high levels of inflammation, but the literature suggests that about 25 to 45% of patients with MDD will have it, and what's specifically shown in the graph on the right is that as C-reactive protein levels increase, so does glutamate. And if you remember from the Psychoneuroimmunology Basics Part 1 podcast, which is also available in PsychU as was mentioned, glutamate spillover plays an important role in disrupting communication between neurons, which can alter our behavior and produce some of the symptoms we see in major depressive disorder. In the next slide, the figure on the left shows the correlation between symptoms and the inflammatory cytokine interleukin-6. And this was measured in plasma samples from patients with major depressive disorder. And the graphic that you're seeing there, the larger pieces of the pie, if you will, show the stronger correlations between interleukin-6 and that particular symptom. The strongest observed correlations were for suicidal thoughts and low self-esteem, but significant correlations were also observed for sadness, guilt, concentration, and tiredness. The figure on the right shows the inflammatory cytokine interleukin-1 beta and shows that it's significantly elevated in cerebrospinal fluid of patients with MDD compared to healthy controls. So again, this highlights the idea that the peripheral inflammation that we're measuring in the plasma looks like it is transmitted to the brain, as these correlations are observed for both plasma samples and samples from the cerebrospinal fluid. So I told you earlier that inflammation increases glutamate, but it can also affect other neurotransmitters. So here you see that in suicide attempters, pro-inflammatory interleukin-6 levels correlate with higher dopamine and serotonin metabolism, suggesting lower bioavailability of the neurotransmitters that really are targeted with our current antidepressant treatments. So interestingly, there are multiple ways in which inflammatory cytokines can decrease monoamines like dopamine and serotonin. It involves the pathways that mediate synthesis of these neurotransmitters. In addition, and it's not shown here on the screen, but in the study where this data originated, the authors also reported a positive correlation between interleukin-6 and severity of depressive and suicidal symptoms. In the next slide, we'll talk about how this information potentially might be used in the clinic, although it's not quite there and there's still work to be done to really understand this. It gives us an idea of where things might go in the future. In the top panel, you see results from a study showing that in individuals with major depressive disorder, lower C-reactive protein levels or low inflammatory status, those patients showed greater symptom improvement with an SSRI antidepressant, whereas patients with higher C-reactive protein levels or higher inflammatory status showed greater improvement with a tricyclic antidepressant. So, might be thinking about using some of these strategies of measuring inflammation to determine appropriate treatment strategies. In the bottom panel, the results of a separate study are consistent with this and showed that higher pro-inflammatory interleukin-6 levels are associated with refractoriness to antidepressant treatment. So Rachel kind of touched on all these things, but this is really some of the data behind why the thinking is moving in this direction. And there's another thing to think about here. So we know that there is an established relationship between obesity, metabolic syndrome, depression, and increased markers of inflammation. This study showed that in patients with major depressive disorder and a high body mass index showed slower clinical improvement with antidepressant treatment, and the slowest improvement was observed for patients in the obese category, which is a BMI over 30. And in this study, I should also note that they controlled for the effects of weight, so it's not a dose issue. It has more to do with the obesity itself and how it might be related to the slower clinical response to treatment. So in the next slide, I will tie this together a bit more. Depression and obesity share three biological pathways. On the left there, you see dysregulation of the HPA axis, which may be from early life stress or chronic stress or genetics. The high inflammatory burden that I've showed you a couple of slides now to show the high levels of pro-inflammatory cytokines in major depressive disorder. And then on the left, the dysregulation of energy metabolism. And this brings up an important area that we don't have time to go into for this particular presentation, but insulin resistance, as observed in type 2 diabetics, is associated with impairments in memory and executive functioning, as well as neuronal damage in the hippocampus and prefrontal cortex. So that's an interesting area that would be an interesting presentation to delve into that a little bit more and talk about the mechanisms that are at play there. But taken together, the three pathways really interact to disrupt brain connectivity and promote both obesogenic as well as depressogenic behaviors. So I'm going to turn it back over to Rachel now to discuss the role of inflammation in bipolar disorder.
Thank you so much, Heather. You really gave us a great synopsis about inflammation and depression. So we're going to just keep this train moving and talk about what evidence there is for inflammation in bipolar disorder. So as we see here, we're just getting a synopsis about some of the initial reports dating back to the mid-2000s showing elevated blood levels of pro-inflammatory cytokines, namely IL-8, TNF-alpha, and IL-6, in both bipolar manic as well as depressed individuals compared to healthy controls. And these are folks who had no family history or personal history of bipolar disorder. The evidence since has consistently indicated that bipolar is linked to alterations in cytokine levels. It is interesting that cytokine profiles may actually differ depending on the stage of bipolar disorder. When bipolar patients in a manic and depressive phase are compared to normal controls, a pro-inflammatory state with high concentrations of pro-inflammatory cytokines and low concentrations of anti-inflammatory cytokines have been reported. This elevation in the level of pro-inflammatory cytokines actually seems to fade during euthymic phases, and it suggests that inflammation may be a component of the acute episodes rather than a trait that affects the prospective course of bipolar. Acute mania appears to be specifically linked with pro-inflammatory states, and the data indicate that IL-6 and IL-18 may be markers of manic episodes in rapid cycling bipolar patients. Elevated CRP levels, which we've been hearing a little bit about, are also known to be associated with impaired cognitive functioning, which obviously has an impact on long-term functioning for individuals with bipolar disorder, as well as other disorders like schizophrenia and cardiovascular disease, as well as depression. In addition, evidence for cytokine activity suggests that a fundamental abnormality in oxidative energy generation, high rates of mitochondrial diseases, and mitochondrial DNA mutation may be associated with bipolar disorder. And this could be perhaps related to alterations in antioxidant enzymes, which have also been observed. As we move into the next slide, we'll see a synopsis of some of the typical inflammatory responses, and we'll see here that evidence for an inflammatory response in bipolar disorder actually stems from a multitude of research, with a typical inflammatory response consisting of four principal components that are depicted for you on this slide and labeled out in numbers for you. The table actually summarizes findings across components of the inflammatory response with respect to bipolar, reflecting that evidence across all four of these components has been replicated in bipolar patients. Since not all individuals with bipolar show an increased inflammation or inflammatory tone similar to what we see in depression, it appears to be a subset of the patients. Experts have actually begun to explore the possibility of an inflammatory phenotype among bipolar patients. So as we advance to see our graphic, which is the next component of this slide, we'll actually be looking at some putative mechanisms that underlie the immune dysfunction within individuals with bipolar. Evidence suggests that bipolar is associated with a diminished cellular resilience and higher levels of apoptosis. This may lead to the release of damage-associated molecular proteins, or DAMPs, from dying cells. These DAMPs have been shown to be elevated in blood from bipolar patients, and they can activate and trigger a cascade involving the inflammatory response. We know that overactivation of microglia and an imbalance between pro and anti-inflammatory microglial phenotypes can lead to higher levels of inflammatory cytokines and really keep in motion this feed-forward activation loop of dysregulation in microglial cells. In addition, the HPA axis dysfunction and hypocortisolemia that's been reported in bipolar disorder patients may also result in an imbalance in production of pro- and anti-inflammatory cytokines. And this could arise from changes and dysfunction at the glucocorticoid receptors, which Heather has told you a little bit about. In addition to HPA axis irregularities, bipolar is also associated with excess sympathetic nervous system activity. And experts are actually suggesting that this constellation of sympathetic nervous system overactivity, parasympathetic withdrawal, glucocorticoid receptor insufficiency, and elevated inflammatory signaling can help account, at least in part, for the increased risk in metabolic syndrome, endocrine disorders, and vascular diseases that we see in bipolar disorder. As Heather was just mentioning, there's a similar reflection in our major depressive disorder patients. When we stop to think a little bit about the growing realization that major mental health disorders are fundamentally progressive, we know that simpler treatment requirements and a better prognosis typically is required in the initial stages of the illness. And this has led a lot of experts and clinicians to start thinking about staging of some of these disorders. So it must be kept in mind that a staging approach actually has been highly beneficial in the overall management of other complex disorders, including diabetes, cardiovascular disease, and various forms of cancer. This actually just presupposes that illness advances in an identifiable temporal progression of phases from an at-risk population into a prodromal period, where the first clinical manifestations reveal themselves, and then further developing into more severe, intractable forms with evolution and chronicity of the illness. So this term neuroprogression has been increasingly used to define the pathological reorganization of the central nervous system, which arises as a result of many different types of insults, environmental as well as genetic and epigenetic, including things like stress and oxidative stress and inflammation. And you can see some of the different clinical underminings and clinical findings that can be associated with this, as well as the biological findings on the right portion of the slide. Essentially, the experts are thinking that a mounting of allostatic load can be set in motion to really perpetuate illness progression through various stages, making it more and more difficult to treat some of our patients with mood disorders, particularly bipolar patients. We're also going to talk just for a moment about inflammation and neurocircuits. So inflammation is increased in patients with mood and anxiety-related disorders due to various different types of environmental challenges, diet, lifestyle, things that we've talked about, as well as medical comorbidities and psychosocial stress. This can alter their inflammatory tone, as we've been discussing all along, and actions of inflammatory cytokines ultimately contribute to alterations in neurocircuits within the brain. These include structures related to basal ganglia to prefrontal reward systems and motor circuits, as well as those that mediate fear and anxiety-related behavior from the amygdala, prefrontal and insular circuitry, and all of these can contribute to presentation of symptoms like depression and anxiety. On a cellular level, the immune system can detect danger signals in the absence of a pathogen, something we've evolved to do. And this is related through the release of danger-associated molecular patterns or damage-associated molecular patterns, such as the DAMPs that we talked about just a few minutes ago, which can set in motion a cascade of events and processes that have been termed sterile inflammation. So there's no pathogen there, but we're detecting something that we think is a perceived threat in the environment, which could be psychosocial stress. And this can lead to a very similar inflammatory process as if there were a saber-toothed tiger or a viral infection that would have actually induced the same type of inflammatory change. So the take-home message here is that there's evidence from peripheral inflammation suggesting that it affects brain regions that are relevant to reward and threat sensitivity, and this can actually lead to symptoms of reduced motivation motor slowing, and anxiety, all consistent with what we see in patients with mood disorders and depression and anxiety. When we think about inflammation and its impact on cognition, hopefully it has been clear through some of our previous discussion that it makes sense cognition may be affected by an increase in the inflammatory tone. There's been a recent publication by Furrier and colleagues that assessed evidence for inflammation-mediated cognitive impairment across major depressive disorder, bipolar disorder, schizophrenia, and post-traumatic stress disorder individuals. We know that increased peripheral inflammation is associated with many of the characteristic symptoms of mood disorders and anxiety disorders. I won't list those again now. You've heard them so many times at this point. But cognitive deficits, including things associated with attention, verbal learning, and verbal memory, which are commonly associated with all of these different disorders, they usually predict a poor general and psychosocial outcome or functioning for patients, as well as impair recovery in later stages of the illness. So what we're looking at here with the brain image, we see a role of neuroinflammation in the dysregulation of neurobiological processes that underlie cognition. The numbered items that are shown here, you see that 1 through 5. As we walk through the graphic, we notice that neuroinflammation induces dysregulation of various neurobiological mechanisms, and that can impact cognition in each of these different 5 ways. So the first one is showing you changes at the expression and activity level of AMPA receptors, one of our glutamatergic types of receptors, and this can impair homeostatic plasticity. We also know, if you look at number 2, that there's inhibition in long-term potentiation and long-term depression, which can ultimately impact neuroplasticity and our ability to learn and gain new memories. Number 3 is showing us dysregulation of the tryptophan kynurenine pathway, which Heather's talked to you a little bit about. So this alters our availability of various monoamines, namely serotonin, but we know this is true for others as well. And this can actually subsequently lead to some neurodegenerative processes. 4 is showing you impairment at the neurotrophin level, which we know is imperative for maintaining happy and healthy brain cells, glia, and neurons. And then number 5 is showing you dysregulation at the level of the HPA axis, which can potentially lead to hypocortisolemia, as well as some subsequent neurotoxicity. All of these are involved in neuroplasticity, as well as overall functioning of our cognitive processes. As we advance to look at the next graphic, this ties it home a little bit more for our clinical ramifications of what's going on here. So this actually shows you, if you look in the middle portion, all of the different aspects of cognitive domains that are impacted by an increased inflammatory tone. And then you can see how this might actually impact our processing speed, as well as our ability to interact in social environments. And we know that this common pattern of cognitive alterations that are seen across so many psychiatric disorders actually increase the severity of symptoms, decrease the quality of life and workplace, as well as psychosocial functioning for individuals who are suffering from these illnesses, as well as just having an overall negative effect on remission and recovery, as well as an increased risk for relapse. So the summary for cognition and inflammation is really that there is a common pattern across many psychiatric conditions, as we've described here, and this raises the question as to whether or not the inflammatory process, as it disrupts neurobiological mechanisms, can be a common underlying pathway and something that we can target for treatment to improve outcomes for individuals with many of our psychiatric diagnoses. Another interesting area that is really hot topic at this point in time is looking at the interaction between the bugs in our gut, if you will, as in the brain, and how this can lead to development of mood disorders. The human gut is home to numerous bacteria, where the gut bacteria outnumber human cells at least tenfold. This is known as the gut microbiome, or this environment of various bacteria within the gut. And they facilitate nutrient absorption by metabolizing indigestible dietary compounds, as well as developing gut-associated lymphoid tissue or gall. to produce vitamins and prevent colonization of pathogens within the level of the gut. So involved in that innate immunity for sure, and have a role in our adaptive immunity as well. The composition of the gut microbiome correlates with host genetic variation in immunity-related pathways. We mentioned in the PNI basics deck that about 70% of our immune cells actually live, born, and thrive within our gastrointestinal tract. It has been hypothesized that dysfunction in gut flora can lead to states of chronic and acute disease, and we're growing to appreciate this more and more across human health as well as human illness. If you look at the graphic, this is giving us a schematic representation of the microbiota gut-brain axis. There are at least 5 pathways involved in communicating between the microbiota and the brain. And these include the neuroanatomical pathway, which is represented by spinal efferent neurons via the vagus nerve, the neuroendocrine or HPA axis, intestinal and systemic immune activation. It can lead to altered permeability of the intestinal epithelium and the blood-brain barrier. And finally, microbial production of neurotransmitters such as GABA and serotonin. As we are aware, the blood-brain barrier exerts tight regulation over movement of ions, molecules, and cells between the central nervous system and the blood. And a disruption in the components of the gut microbiome can actually alter not only the permeability of the gastrointestinal system and the gut itself, but also can alter permeability of the central nervous system, or what gets into and out of the brain. At the very bottom, you'll notice there's implications for probiotics and prebiotics. So probiotics are consumed microorganisms that are intended to alter host gut microbiome and provide health benefits, whereas prebiotics are consumed dietary fibers that are meant to improve health by feeding and promoting balanced intestinal microbial ecosystem. Both of these avenues are avidly being pursued and looked at for intervention for a number of different types of human illnesses, including mood disorders. Zonulin is on the left portion of the slide with a question mark beside it. This is being appreciated more and more these days as a potential biomarker for dysbiosis in individuals who have psychiatric disorders. So it's essentially a protein that is highly involved with maintaining tight junctions between the duodenum and the small intestine. So it really regulates whether or not that permeability of the gut is maintained as it should be. And we notice that there's individual differences, particularly in those with mood disorders, with their expression of zonulin. So we are looking at this as a potential treatment option. Overall, this graphic is intended to provide an integrative model that details the mechanisms whereby inflammation, oxidative stress, mitochondrial dysfunction, bacterial translocation, and dysbiosis, as well as neuroinflammation, may cooperate to cause and maintain permeability of the blood-brain barrier and leading to different types of neurodegenerative, neuroinflammatory, and neuroprogressive diseases. So finally, I'm just going to try to wrap all of this up in terms of what we've talked about for bipolar disorder. And hopefully I've truly left you with the sense that this could potentially be a multi-system disorder that we need to think about staging. We need to think about the course of illness and potentially individualized treatment based on that. And overall, experts are actually proposing that the common inflammatory processes could potentially underpin both bipolar disorder as well as many of the common comorbidities that we see, including cardiovascular disease, hypertension, some of the things depicted on the slide here. They propose that in the modern conceptualization of bipolar disorder as a multi-system disorder could really change the way that we think about long-term treatment with medications as well as lifestyle interventions, and hopefully lead to an overall better prognosis for bipolar patients as we grow to appreciate this multi-system illness a little bit more with growing scientific evidence. So with that, I'm going to transition over to Carolyn to continue our discussion today about potential anti-inflammatory treatment approaches.
Thanks, Rachel, and thank you to you and Heather for that excellent overview of our current understanding of the science of the psychoneuroimmunology of mood disorders. So now let's look at the treatments. So there are several different anti-inflammatory treatment approaches that are currently being studied for mood disorders, and these include both pharmacologic and non-pharmacologic treatments. So let's start by reviewing the pharmacologic approaches. The most frequently investigated type of drug in clinical trials for mood disorders are the non-steroidal anti-inflammatory drugs, or NSAIDs. And a meta-analysis of 14 clinical trials suggested that NSAIDs do promote improved antidepressant effects compared to placebo, but the efficacy data for specific depressive symptoms is still lacking. There are two main types of NSAIDs, so the selective and non-selective COX inhibitors, both of which have been shown to have an antidepressant effect. For the selective COX-2 inhibitors, they're thought to have a larger anti-inflammatory and antidepressant effect compared to other NSAIDs, and they have been observed to have efficacy in both MDD and bipolar disorder. The non-selective COX inhibitors are readily available and inexpensive, but there are currently no randomized controlled trials as adjunctive treatments, and the monotherapy trials have shown mixed results. So we continue to need more research in this area of the NSAIDs. Other types of pharmacologic treatments that have been studied are cytokine inhibitors and tetracycline antibiotics. As their name suggests, cytokine inhibitors have a direct anti-inflammatory effect through their blocking of specific cytokines. 4 monotherapy trials have shown borderline significance towards a better antidepressant effect than placebo. The most commonly studied drugs in this class are the anti-TNF therapies, which have shown a significant antidepressant effect. Interestingly, these drugs may not actually enter the brain, but may exert their effects through targeting peripheral inflammation. Conversely, tetracycline antibiotics readily cross the blood-brain barrier and are proposed to have antidepressant effects through their neuroprotective activities. Small trials have shown significant improvement in depressive and bipolar symptoms, and clinical trials are currently ongoing for these compounds. Several other pharmacologic anti-inflammatory treatments for mood disorders have been proposed, but their efficacy has not been extensively researched. These include N-acetylcysteine, coenzyme Q10, steroids, neurosteroids, melatonin, polyunsaturated fatty acids, vitamin D, and specific anti-diabetic drugs. One outstanding question about anti-inflammatory treatments is related to the timing and duration of treatment required for efficacy. Long-term use of anti-inflammatory agents is associated with side effects, so there's a question about the potential risk-benefit ratio for their prolonged use. Current studies suggest limited use for mood disorders in the acute phase is sufficient for efficacy, and this limits exposure-related side effects. A variety of non-pharmacologic anti-inflammatory treatment options have been suggested and researched. These include caloric restriction and intermittent fasting, cognitive behavioral therapy, enhanced nutrition, exercise, increased socialization, meditation, mindfulness, phytoncides, probiotics, prebiotics, and symbiotics, as Rachel talked about, and sleep. Further research is required to determine their efficacy and their synergy with pharmacologic interventions for mood disorders. In summary, the etiology of mood disorders is a complex phenomenon arising from multiple genetic and environmental influences, of which neuroinflammation is now thought to be a significant contributing factor. The complex pathophysiology composing these disorders results in an equally complex clinical presentation, but also gives the opportunity to identify subtypes of these disorders to increase treatment efficacy. Neuroinflammation has been proposed to be a contributor to this pathophysiology, and further research is required to investigate anti-inflammatory therapeutic interventions. That concludes our overview of the psychoneuroimmunology of mood disorders. I'd like to thank Rachel Self and Heather Davidson for joining the presentation today. And if you have any further questions in relation to the material presented today, please follow up with your local MSL. Thank you for joining.